Hello students, welcome to readmedprep Academy channel. Today in Applications of Biotechnology Part 4, we are going to talk about the transgenic animals, biological products, animal cloning, ethical issues of cloning. First, let us move on to the transgenic animals. In early days, selective breeding methods were carried out to improve the genetic characteristics of livestock and other domestic animals. With the advent of modern biotechnology, it is possible to carry out manipulations at the genetic level to get the desired traits in animals. What is meant by transgenesis? It is the process of introduction of extra, that means foreign or exogenous, DNA into the genome of the animals to create and maintain stable heritable characters. What are GMOs? The foreign DNA that is introduced is called the transgene, and the animals that are produced by DNA manipulations are called transgenic animals, genetically engineered animals, or genetically modified organisms called GMOs. First transgenic cow in 1997, Rosie, the first transgenic cow, produced human enriched milk which contained the human alpha lactalbumin, and the protein rich milk, about 2.4 gram per liter, was a nutritionally balanced food for newborn babies than the normal milk produced by the cows. The various steps involved in the production of transgenic organisms: first is identification and separation of the desired gene, selection of a vector, generally a virus, or direct transmission, combining the desired gene with the vector. Number 4, the introduction of the transferred vector into the cells, tissues, embryo, or mature individual. Number 5, demonstration of integration and expression of foreign gene in the transgenic tissue or animals. Number 6, transgenic animals such as mice, rat, rabbit, pig, cow, goat, sheep, and fish have been produced. Here you can see method 1, method 2. In method 1, the recombinant DNA is introduced into the embryonic stem cells, and they select the transformed embryonic stem cells that contain the desired gene. Inject the transformed embryonic stem cells into the inner cell mass. This inner cell mass, which is present in the blastocyst, is introduced or implanted in the uterus of the female, and the foster mother bears the offspring, and the testing is done for the transgene in the offspring and mate heterozygous offspring to produce homozygous transgenic strain. In method 2, the desired gene with the vector is introduced directly into the fertilized egg, and this fertilized egg is implanted into the uterus of the foster mother, and the foster mother offspring produced is tested for transgene, and this mating is done of the heterozygous offspring to produce homozygous transgenic strain. Here you can see the production of transgenic animals, where a egg donor can be taken of the goat or the sheep, and the human gene is taken and is introduced into the egg, and the microinjection of the human gene is done into the goat or sheep, and the development within a host goat is done, and the transgenic goat that is obtained produces milk which is more nutritious. Use of transgenesis. Transgenesis is a powerful tool to study gene expression and developmental processes in higher organisms. Transgenesis helps in the improvement of genetic characters in animals. Transgenic animals serve as good models for understanding human diseases, which help in the investigation of new treatments for diseases. Transgenic models exist for many human diseases such as cancer, Alzheimer's disease, cystic fibrosis, rheumatoid arthritis, and sickle cell anemia. Transgenic animals are used to produce proteins which are important for medical and pharmaceutical applications. Transgenic mice are used for testing the safety of vaccines. Why use transgenic mouse models? The development of human diseases can be done in a controlled environment. Test possible new drug treatments and get faster results. Target specific genes to study. Replicate specific characteristics, symptoms, or pathology of the disease. Transgenic animals are used for testing toxicity in animals that carry genes which make them sensitive to toxic substances. Then non-transgenic animals exposed to toxic substances and their effects are studied. Transgenesis is important for improving the quality and the quantity of milk, meat, eggs and wool production in addition to testing drug resistance. Here you can see the transgenic goat cells with gene for human growth hormone, and the microinjection of these 2N nuclei into enucleated donor eggs got from the donor. And the development is done within the host goats of the offspring. And the offspring or the clone transgenic goats that are produced have production of better human meat and better human growth hormone. The clone transgenic goats produce human growth hormone which can be extracted from the milk. Now let us move on to the biological products and their uses. What is a biological product? A biological product is a substance derived from a living organism and is used for the prevention or treatment of disease. These products include antitoxins, bacterial and viral vaccines, blood products and human extracts. The examples of biologics from the somatic cells, gene therapy, vaccines, recombinant therapeutic proteins, allergenics, blood and blood components and various types of tissues extracted. These products may be produced through biotechnology in a living system, such as a microorganism, plant cell or animal cell, and are often more difficult to characterize than small molecule drugs. Through recombinant DNA technology, it is possible to produce these biological products on demand. Here the source materials, it can be the mice, the humans, the mammalian cell cultures, the yeast, the transgenic animals, bacteria, mycoplasma, fungi, viruses and TSE agents. There are many types of biological products approved for use. They are therapeutic proteins, monoclonal antibodies and vaccines. Healthcare and pharmaceutical industries have been revolutionized by biotechnological proteins. Hormones and antibodies are produced commercially, primarily for the medical industry. Recombinant hormones like insulin, human growth hormone can be done. Here you can see the production of recombinant vaccines where the whole inactivated vaccine can be produced, or live attenuated vaccine can be produced, or synthetic peptides can be extracted for vaccination. Recombinant subunits can be used as vaccines. Recombinant viral vectors can be used. Recombinant bacterial vectors or DNA itself can be used directly as a vaccine. Recombinant proteins production like human alpha-lactalbumin are also available today, where the DNA construction is done of the plasmid with the required gene and a cell transfection is done. Cell propagation and protein production is done, and the cell lysis occurs which results in the protein purification and extraction of the protein. Animals are used as bioreactors to produce desirable proteins. Antibodies are substances that react against the disease causing antigens, and these can be produced using transgenic animals as bioreactors. Monoclonal antibodies, which are used to treat cancer, heart disease and transplant rejection, are produced by this technology. You can see here the recombinant antibodies production, where immunization is done to the animals. FACS based B cell selection is done. Positive B cell screening and single clone identification. Validation is done, and PCR amplification, where the light chains and the heavy chains are produced in large numbers, and the light chains and the heavy chains are incorporated with the plasmids, and cloning is done with the plasmids, and the recombinant antibody expression is done in the mammalian cells, and a purified antibody is extracted from these. Natural protein adhesives are non-toxic, biodegradable and rarely trigger an immune response. Hence could be used to reattach tendons and tissues, fill the cavities in teeth and repair broken bones. Now let us move on to animal cloning. What is meant by cloning? Cloning is the process of producing genetically identical individuals of an organism either naturally or artificially. In nature, many organisms produce clones through asexual reproduction. Cloning in biotechnology refers to the process of creating copies of organisms or copies of cells or DNA fragments. That is called molecular cloning if DNA fragments are produced. Dolly was the first mammal sheep clone developed by Ian Wilmot and Keith Campbell in 1997, whose images are given below. Dolly, the transgenic clone, was developed by the nuclear transfer technique and the phenomenon of totipotency. Totipotency refers to the potential of a cell to develop different cells, tissues, organs, and finally an organism. The mammary gland udder cells or the somatic cells from a donor sheep or Eve were isolated and subjected to starvation for 5 days. The udder cells could not undergo the normal growth cycle. They entered into a dormant stage and became totipotent. An ovum or egg cell was taken from another sheep, Eve, and its nucleus was removed to form an enucleated ovum. The dormant mammary gland cell or the udder cell and the enucleated ovum were fused. The outer membrane of the mammary cell was ruptured, allowing the ovum to envelop the nucleus. The fused cell was implanted into the another Eve, which served as a surrogate mother. 5 months later, Dolly was born. Dolly was the first animal to be cloned from a differentiated somatic cell taken from an adult animal without the process of fertilization. Here you can see in the image how the cloning method is done. First, the somatic udder cells is taken and it is allowed to starve, and dormancy of the udder cell is done, and an egg is taken from an Eve, and the fusion of the udder cell nucleus and the egg cell nucleus is done, and the rupture of the membranes are done, and this is implanted into an Eve, and slowly these cells develop into a blastocyst and slowly become organized and form a full organism, and this was done in Dolly cloning. Ian Wilmot and Campbell removed 277 cells from the udder of an adult sheep and fused those cells with 277 unfertilized egg cells from the nuclear material was removed. After culturing the resulting embryos for 6 days, they implanted 29 embryos into surrogate mother's womb and only one Dolly was produced. What are the advantages and disadvantages of cloning animals? It offers benefits for clinical trials and medical research. It can help in the production of proteins and drugs in the field of medicine. Aids stem cell research. Animal cloning could help to save endangered species. Animal and human activists see it as a threat to biodiversity, saying that this alters the evolution, which will have an impact on the populations and the ecosystem. The process is tedious and very expensive. It can cause animals to suffer. Reports show that animal surrogates were manifesting adverse outcomes, and cloned animals were affected with disease and have high mortality rates. It might compromise human health through consumption of cloned animal meat. Cloned animals age faster than the normal animals and are less healthy than the parent organism, as discovered in Dolly. Cloning can lead to occurrence of genetic disorders in animals. More than 90% of cloning attempts fail to produce a viable offspring. What are the advantages and disadvantages of cloning summed up here? Advantages: solution to infertility, provides organs for transplants, genetic modification, healthiness of infants, better understanding of genetic diseases, helps improve life. Whereas disadvantages: uncertainty of science and technology, losing diversity in genes, the diseases leading to distinction, religious problems, transgressing nature, inhumane act. Now let us move on to the ethical issues. Biotechnology has given to the society cheap drugs, better fruits and vegetables, pest-resistant crops, indigenous cure to diseases, and lot of controversy. This is mainly because the major part of the modern biotechnology deals with the genetic manipulations. People fear that these genetic manipulations may lead to unknown consequences. The major apprehension of recombinant DNA technology is that unique microorganisms, either inadvertently or deliberately for the purpose of war, may be developed that could cause epidemics or environmental catastrophes. Although many are concerned about the possible risk of genetic engineering, the risks are in fact slight and the potential benefits are substantial. The moral and the legal issues of cloning. Do people have a right to reproduce by any available means? Do other societal concerns override any such rights? Will there be a harmful effects on the clone twin? How will family relationships be redefined? Could persons be cloned without their consent? Would cloning be immoral because it is unnatural? Though human cloning is unethical, cloning organs or body parts for therapeutic or medical purposes is ethical. Today, in Applications of Biotechnology Part 4, we discussed about the transgenic animals, biological products, animal cloning, ethical issues of cloning. So thank you. Kindly subscribe, like, share and comment the channel Read MedPrep Academy.